All right, good afternoon. You can all hear me sufficiently? Fantastic, good. All right, so as Norman said, I am Jamie Ellis. I work for the University of Florida. I always start with my title slide. So the University of Florida is a fairly large institution. We have 55,000 students in the University of Florida. I work in the Entomology and Nematology Department. Entomology is the study of insects. Nematology is the study of nematodes, which are microscopic worm-like creatures. You can see some of them, but most of them are really tiny, so you have to look at them with a microscope. We have relatively few nematologists in our department, so most of us are entomologists. And as I was sharing with the audience yesterday, our department has about 80 professors in it, so we're a really large entomology department as entomology departments go. So I am talking to you today about the mating biology of Western honeybees. This is stuff that's generally true across the species. Biology is incredibly messy, so you can get lots of variation and variety mixed into this species, but I'm going to tell you some generalizations related to the mating biology of Apis mellifera, the Western honeybee. I won't even spend time on talking about how you correctly spell honeybee. That's another battle for another day. But the fact that I have two words on my slide tells you what the right answer is, doesn't it? But I know the British do something different. Europeans do as well. All right. Now, there's a lot of text on this slide. I appreciate that. But as I was sharing with the audience yesterday, the science of teaching is called pedagogy. And in pedagogical research, the worst possible way to teach people something is through lectures. So that means I am currently teaching you the worst possible way you can learn. So what I am going to do is put up QR code here so that long after this meeting is over, you can go to that QR code and go to a document that has a very similar name that's at this website, and you can read everything I'm going to tell you this afternoon and then some. If you still have an insatiable appetite for the mating biology of honeybees, there is a book that's similarly titled that I co-authored with Nico and Gudrun Kerniger and Larry Connor, and you can go buy it. It makes great bedtime reading. It's a great book to have anywhere you want. All right, so because I'm lecturing to you, I want to tell you now what I hope you learn through this lecture, and I'm going to remind you at the end that I taught that to you, okay? All right, so by the time we're done, I hope you will understand some key drone and queen organs that mature before and after they emerge from their cells. You'll recognize some characteristics of DCAs, or drone congregation areas. You'll know a bit about how DCAs help promote genetic diversity. We'll talk about honeybee copulation, and I have to use exclusively science terms because my 10-year-old daughter's in the room. So this is the lecture that she attended of all the ones that I'm giving. All right, indicate the benefits of polyandry to honeybees and recognize how honeybee queens mature after mating. If you're struggling with the science terms, see me after and I'll use the local slang, okay? Maybe I won't. I don't use those words, even in my regular words. All right. So all of you know that honeybees have a really remarkable sex determinant system. That means their sex is determined in a relatively interesting way compared to our sex determinant system. In honeybees, you know males are haploid, females are diploid. Now, we think that that's interesting, but it's actually not unique to bees. All of you went through biology at some point in your schooling, and you'll remember there's kingdom, phylum, class, order, family, genus, species. Right? You all know that. And somewhere around class Insecta, kingdom, phylum, class, order, there are multiple orders of insects. Honeybees belong to an order of insect called Hymenoptera. Hymenoptera in the Greek means membrane, hymen, optera wing, membranous wing. So it is an order of insects whose members have membranous wings, which includes the bees, the wasps, the ants, and the sawflies. All Hymenoptera determine their sex this way. We just happen to study honeybees and keep them in our boxes so we can harvest their vomit for sale. But all Hymenoptera have a sex determinant system where the males result from unfertilized eggs and the females result from fertilized eggs. That's pretty remarkable if you think about it. So an unfertilized egg will produce a male, a drone. A fertilized egg will produce either a queen or a worker, depending on the quality and quantity of diet it is fed while she is developing as a larva. There's some recent research that I've had to talk a lot about because once people found this research, they kept asking me questions. There is some compelling evidence at the moment that queens will lay slightly larger eggs into queen cups than they lay into worker cells, and that the resulting queens are better than queens that are reared from eggs that were deposited into worker cells. So that's led to some speculation that there could be royal eggs, but that's another talk for another day. For the purposes of discussion here, I'm going to say fertilized eggs, which become female, either well-fed or not well-fed, become queens or workers. Are you still with me? Yes. Fantastic. That's great. And I think that's what everything here says. So we have a haplodiploid sex determinant system present in honeybees. It's quite remarkable, and this is how it works. Now, I will tell you, there are, again, 30 to 35 different subspecies of Apis mellifera. Most of the time, unfertilized eggs result in males. But there is one subspecies of Apis mellifera, when she lays an unfertilized egg, that often results in females. Do you know what that subspecies is? It's trivia time. Capensis, you are correct. It is Apis mellifera capensis. That is the Cape honeybee of South Africa. When their colonies go queenless, their workers can become laying workers, and when their workers lay unfertilized eggs, a large percentage of those eggs result in females, even though the laying worker could not mate and fertilize that egg. You knew that, right? The process is called Thelotocus parthenogenesis. It just rolls right off the tongue, right? Parthenogenesis is essentially the act of an unfertilized egg producing an offspring. So parthenogenesis happens even in the process of producing drones. But that is called arenotocus parthenogenesis. So when it produces a male, it's arenotochy. When it produces a female, it's called thelotochy. When you're a beekeeper, who cares, right? Right? So let's move on. I just thought I'd tell you that because some of you want a little bit more. And I will tell you, every time I go to the UK or Ireland or other beekeeping places in this part of the world, you always ask the hardest questions on planet Earth. So I'm telling you some sciencey stuff for those of you who are gearing up to ask me those questions I cannot answer. All right. So now this has to happen somehow. Right? This has to happen somehow. There's got to be some biology that occurs that produces these haploid males or these diploid females, and that is what I'm going to discuss in this talk. So, when you get immature drones and immature queens, they are already heading in the direction of being fertile individuals, right? You've got the production of queens that are developing these queen cells. You've got the production of drones that are developing in these drone cells, and there are organs that begin to develop while these individuals themselves are developing or are immature. So let's talk about that for just a little bit. We know that when eggs are laid into a queen cell and that resulting female larva is being treated as if she were going to be a future queen, the entire length of time it takes for that egg to go to adulthood is 16 days. That adult queen takes 6 additional days. The adult queen takes 6 additional days to mature sexually fully, which is why she doesn't come straight out of the cell and go straight on her mating flights. She needs about another week to fully develop sexually before she can go out and take her mating flight. So the entire length of time is somewhere in the neighborhood of 22 days from egg to being able to mate. For drones, that period is somewhere in the neighborhood of 36 to 44 days. We know from egg to adult drone, it's 24 days, so it's an extra week or 2 weeks for that drone to mature sexually and be ready to go on these mating flights. So let's talk a little bit about queens. When queens emerge from their cells, as I've shared, they are not ready to mate. They're not even really treated by other workers in the nest as if they are queens. They might be fed a little bit, but queens do a lot of their own meandering, feeding themselves. We know that they are small. They're virgin queens. We recognize them as smaller queens. And the reason for that is mating triggers the fleshing out of the ovaries in that queen, so she gets bigger after she mates and in the process of right before she mates. So she is, as a young virgin queen, running around the nest. She's soliciting for food occasionally from worker bees. She's not treated hostilely, but she's not treated as she will in the future be treated once she is mated. She won't command that attention that you see worker bees typically give fully mated queens. Drones, on the other hand, when they are coming out into the world, have to be taken care of by their sisters because they're lazy bums who can't do anything for themselves, right? There's no parallels between humans and drones. There's no equivalent in the human world. But in the bee world, drones are lazy bums, and so their sisters take care of them when they emerge from their cells, whereas queens have to do a little bit on their own before they end up going on that mating flight. I will tell you, my favorite sentence in this whole presentation is that last one. Young drone activity is largely limited to resting, eating, begging for food, and combing their hair, right? Right? That's pretty much what the young men do before they go out on their mating flights. And they have to do this because they're not ready to go on their mating flights. Their flight muscles are not mature enough to fly. So they have to spend some days developing before they are, in fact, capable of flying. The same is true, incidentally, for queens and even for workers. Workers don't come out of their cells ready to fly. They're incapable of doing that. Early on, queens may go through short orientation flights. Someone emailed me a couple of weeks ago and said, Jamie, do queens take orientation flights? So I had to spend a few days searching through the literature. I mean, I knew they did, but I wanted the research to back it up. I found the research to support the fact that they do take orientation flights, which is pretty intuitive because if they went on a mating flight without knowing where home was, how would they know how to come back? Women just know this kind of stuff, right? But the drones would never make it back to colonies if there weren't an orientation flights involved, right? They need some sort of directions when they go out and come back. And so you see queens and drones starting their orientation flights somewhere about a week after they emerge as adults. Now let's talk about some of the internal anatomy of queens and drones. I find this amazing. Actually, Norman and I co-authored with a few other folks a chapter on honeybee anatomy and dissection for the Colos Bee books. And when we were going through the Colos Bee books chapter on honeybee anatomy, I went through all of the Dade images from the IBRA publication, and we were cataloging all the Dade images, and we were saying what organs were what and everything like that, and that is when I learned absolutely the most about internal anatomy of honeybees. You see before you the inside of the abdomen of normal workers, laying workers, and virgin queens. And they have a few things in common. First, they have, at least even the normal workers, have atrophied ovaries. This is where their ovaries would be. Queens have, even virgin queens, have much larger ovaries, waiting to develop fully. We'll talk about that subsequently. And laying workers, you see, have somewhat developed ovaries compared to normal workers, but nothing near what virgin queens have. Another thing that I will point out looking here is that this sphere, which we're going to spend a lot of time on throughout this presentation, is the Spermatheca. This is the organ that holds the semen that the queen receives from drones while she is on her mating flight. Notice that that Spermatheca is absent in workers. That's why they cannot mate and lay fertilized eggs. They cannot receive and hold semen. So you get normal workers, laying workers, and virgin queens. And I'm going to then scale into this and talk a lot more about what you see here. So first things first, the Spermatheca. Some things in anatomy world just make sense. If I said there was an organ called a Spermatheca, I bet you could guess what it does, right? So this organ here is the Spermatheca. It is supported by glands that provide nourishment to the spermatozoa that live in that Spermatheca. It is also surrounded by a tracheal net. If you are a fisherman or a fisherwoman, you know the cast net that you throw out. Imagine that cast net wrapping around a really large ball. The ball would be the Spermatheca. The cast net would be the tracheal system that goes around that Spermatheca. That tracheal system is necessary because that sperm in the Spermatheca must remain alive in order for that queen to fertilize eggs the length of her life. So that sperm in the Spermatheca has two needs. It needs oxygen and it needs energy, and those items are supplied by the tracheal net or the glands, respectively. If you look closely here, that Spermatheca has a duct, D-U-C-T, which is just a tube. That tube goes from the Spermatheca to the vaginal opening, and it's that tube through which sperm is passed to fertilize that egg. It is flanked by a series of muscles around this valve and pump, which allows the queen to withhold semen or release semen as that egg is passing through the chute. Someone years ago asked me, I think it may have been at one of these meetings actually, asked me, is the queen sensitive enough to release just one sperm cell at a time? And I did what any good scientist who didn't know what the answer was said. I said, of course. And guess what? I was wrong. I went back and did the research, found that person, corrected my answer with that person, but now you get the benefit of knowing the answer on the front end without me looking like a fool in front of all of you, which is the queen doesn't release a number of sperm, she releases a volume of sperm. And usually in that volume, there will be one or two sperm cells. You might have more, it's biology. One of my favorite sayings is that biology is messy. So you might have more or fewer, but when the egg passes the chute, she's releasing a volume that will usually have enough sperm to fertilize that one egg. You only need one viable sperm. This is going to be super duper important as we go through this presentation near the end and we talk about queen failure. So queens are just releasing volumes to fertilize that egg. She has no concept, as far as we know, if there's viable sperm in that volume or not. She's just releasing the volume. And all of that happens through the pump and the valve that ends up releasing that volume down the spermathecal duct. Sue Colby, who is a master at artificial insemination, took these amazing images of spermathecae. This one that looks like a crystal ball is from a virgin queen. It is a Spermatheca that has no semen in it. She's holding it on the tip of her finger. That was the tracheal net that was covering that crystal ball, and she teased that tracheal net away from that crystal ball, and you can see that it is empty. So this is a virgin queen. This is a poorly mated queen. Notice in the well-mated queen, you get that marbled appearance. It's got that marbling look to it. This is a well-mated queen. Contrast it with this that does not have that marbling in the Spermatheca. And you can clearly see here the tracheal net around this Spermatheca. Now, once Sue takes these things out, determines the quality of her queen, she puts them back in and puts, no, I'm just kidding. No, she doesn't do any of that. Once you take the Spermatheca out, it's over. That's it. You know what the queen was. She'll never be the same. All right. So this is what you get. Now, let's go back to those fantastic dade images. Let's see here what we've got. You probably recognize from this bulb here that that's the Spermatheca, right? All right, you recognize then that duct right there would be the spermathecal duct. Well, where do the eggs come from? Well, it should be obvious to you. There are two large pear-shaped organs we call ovaries. So this is one ovary, that's the second ovary. And I'm a hand talker, so I'm going to have to put the remote control down and show you some things with my hands. If you look at one ovary, just take the ovary on the left. You will notice that ovary is made of many tubes, right? They almost look like links of sausages. Those tubes, what are they called? Ovarioles. So basically, an ovariole is a link of developing eggs, where the immature eggs are created at the top, and as that immature egg goes down the ovariole, the egg is accompanied by cells called nurse cells that provide nourishment to that egg, so that by the time the egg is at the very bottom of their respective ovariole, they are fully developed and ready for release into the wild. The average queen has somewhere between 125 and 150 ovarioles per ovary. So if you've got 2 ovaries, and you've got, let's just say for the sake of argument, 150 ovarioles, and you've got about 300 eggs ready to be laid at any given time. So as you can imagine, as queens are cranking these things out, it takes a lot of nourishment to come into the system to push out eggs all day long, up to 2,000 eggs all day long. So they're bringing in a lot of nourishment to develop these eggs to make it possible for them to lay those eggs someday. Now what happens? Just like there is a spermathecal duct, there are ducts that are attached to the ovaries. So ovaries are on the side of the body, and if you've taken any biology, you know that the term for the side of an organism is lateral. So this ovary has its lateral oviduct. This ovary has this lateral oviduct. So an egg will pass through here, or an egg will pass through here. The 2 lateral oviducts fuse to form a median oviduct, right? That egg will pass by the opening of the spermathecal duct, and it will be fertilized here or not fertilized here based on the whims of the queen. That's pretty fantastic, right? I mean, that's pretty amazing. So when queens go out on their mating flight, which we'll talk about later in the talk, and come back, their ovaries swell and they become capable of doing everything that I just said. She's pretty remarkable. She's specifically built to lay eggs. That is her function in the nest, one of her principal functions, obviously. Drones, on the other hand, drones, the males, produce all their spermatozoa while they are larvae. Most people don't know that. That has profound implications. That means then, while the drone is developing, the quality of its sperm downstream will be affected by the environmental, nutritional, other conditions that that drone encounters while he is in his cell. So if he's parasitized by varroa, if he is given viruses, if he has poor nutrition, if the environmental conditions are not great, all of these things can affect the number and quality of sperm that he produces. Someone published in an apodology paper in 2020, Lego et al., the actual testes of drones as they develop. So the drones are producing these spermatozoa, these sperm cell, while they are larvae. When they finish being larvae and begin to pupate, you get a different process, right? So here you go. Sperm cell begin developing in the testes. The moment the drone larva hatches from its egg, it stops about 6 days after hatching. That's when they've produced their sperm, period. Now they've got to mature it, grow it, make it ready for use, and they do that as they age. So here again, looking at these amazing data images, you'll see that when drones become adults, their testes atrophy because the sperm migrate from the testes to the seminal vesicles, which we've highlighted in blue here. So if you dissect an adult drone that is sexually mature, his testes have atrophied. The spermatozoa have moved to the seminal vesicles, and they are there waiting for deployment. Now, drones are pretty amazing critters anatomically. We don't give them much credit, but I just want to say this. I mean, that's crazy stuff. Drones have to put the semen that they have stored in their seminal vesicles into queens. so that she can receive it, deposit it into her spermatheca, and then be able to fertilize eggs as she goes. And he does it using his endophallus. I will not untangle that word for you. Endo means inside, phallus means phallus. So it's a phallus stored inside of his body, but it's actually stored inside out in the body. The way that I usually teach this concept is, I don't know if you guys have those clowns here that will blow those long balloons and then twist them into all kinds of weird shapes. All right, so we have those as well. And imagine one of those long balloons that you put your finger on the tip and push the balloon inside out. That is how the phallus is stored inside the drone's body. In order for the drone to evert that phallus in all of its glory, it takes a reasonable percentage of his hemolymph, blood. So the act of eversion paralyzes the drone because he loses so much hemolymph to the deployment of his phallus. Most people think the drone dies in the mating process. He's paralyzed in the docking process. So there are some really important features here. The tip of the drone is where the semen collects. There is a Brillo pad-like structure that I want to point out later, so stay tuned with that. And there are these two just really incredibly intimidating structures that come off of the phallus that are called these horns. And in these cases, it's believed that these help with the docking process, although I think that still needs to be absolutely confirmed. But once the drone everts this, he is paralyzed. He's lost so much hemolymph to his phallus that he no longer is able to move or function. So now we think then about what are some factors that can affect the quality of the sperm that the drone produces. And they're the things that I've mentioned. For example, right? There's all sorts of things that can happen to queens and drones as they develop. Nutrition, environment, pesticide exposure, virus exposure, pathogen exposure, mite exposure, lots of things can happen to impact the quality of queens and drones before they even make it to the drone congregation area. But now that we have got queens and drones that are sexually mature and ready to engage in the mating behavior, they have to go somewhere to do that. This is actually a remarkable biology, a remarkable behavior of insects. Queens do not mate in their nest. There's a logical reason for that. Queens, imagine a situation where this monitor is the hive. This monitor, the colony living in this hive, has lost its queen. They raise a new queen. The queen that has been reared is the sister of the males available in this nest. If she remained in the nest, she would only have her siblings with which to mate. Inbreeding is not good, right? We know that, right? You do know that, right? Inbreeding, no good. All right. It's also no good in the honeybee world. It's okay in the mite world, but that's another story for another day. So queens and drones then have to find a way to pair with other insects that aren't their relatives. And they do this through drone congregation areas. We tend to call these DCAs in the beekeeping world, right? So DCAs are very interesting. They're difficult to study. There's continuing research on them. But essentially what happens is the queen has to go to these DCAs where these drones are congregating. The drones have to find her, mount her, mate, and let's now talk about that process. When they are sexually mature, drones being about 3 weeks old, queens being somewhere in the 7 to 10 day old, they will start flying to DCAs. So let's talk then about DCAs. First of all, how do you study them? Well, it's not easy, because DCAs can occur 30 meters in the air. And they often occur away from our hives. And before queens fly through them, the drones are very loose in a DCA. So you may not even see it as a conglomerate of drones, right? They're just kind of loose, and they're really high and loose, and you don't notice them. When a queen flies through a DCA, the drones will coalesce and chase her, and that's when you know you have one. So how you find one, we did some research with DCAs years ago in my lab. This is my master's student, Ashley Mortensen. She put weather balloons really high in the air with a basket underneath. Those baskets, I actually have it on a slide coming up. Well, here. These baskets you can purchase cigarette filters. The little cotton cubes, the cigarette filters. We paint them black and we put queen pheromone on those cigarette filters. You tie them into these cages and you tie them below the cages, and then you put that weather balloon 30 meters in the air and you just walk around. And you walk around for areas that you think might contain DCAs, and you will start getting drones who find queens by smell. They start zeroing in on the queen using smell, and once they get to the cage, they use their large eyes to locate where the queen is. That's why drones have such big eyes. They smell them to get close, they see them to make the final dock. These black cigarette filters look like queens and smell like queens to the stupid drones. So what you'll do is you'll get drones flying up into this open-ended basket and self-collecting. When drones panic, they just go up. And because they go up, when you find a DCA, the drones self-collect in the basket, and you can go, hey, stupid drone, and you pull the balloon back down, and you can sample all the drones in the basket that have a huge opening that they could escape, but they don't because they're dumb. So you pull it down, you sample them, and that's how you find DCAs. And finding DCAs is how people study DCAs and know that DCAs are usually conglomerates of many, many drones. Thousands upon thousands of drones. There's some estimates in this, 16,000 plus drones from over 200 colonies, potentially in a single area. An individual colony will produce about 2,500 drones for every queen it produces. So there's lots of males in the environment going to these DCAs, waiting for queens to fly through. And this large diversity of drones then lowers the chances that the queen's going to mate with her relative. That's important because if she lays an egg that was fertilized by her brother, the worker bees can detect that inbred egg and they will abort it from the hive. So it's important for her to have genetic diversity because of inbreeding problems, as well as increased genetic diversity is one of the things that made it possible for Apis mellifera to live in Europe, the Middle East, and Africa. Apis mellifera has a natural distribution from the northern part of Europe to the southern tip of Africa. Think about all the climates that it goes through from northern Europe to the Cape of South Africa. That species has been able to do it through polyandry. Many males, queens, mate with many males. It increases their genetic diversity, allows them to conquer environments that they otherwise would not be able to conquer because they've got disease or pest resistance or whatever traits that would help them in a given environment. So they will mate with multiple drones. Let's talk about that process. First, when the weather is good, drones, I'm going to overgeneralize to make it easy for me to discuss, but drones spend about 3 hours a day when they are sexually mature going to, being at, and returning from DCAs. They have roughly 30 minutes of energy in the tank. So when they fill up on honey before they take their flight, they'll fly out to a DCA, do their thing, fly back to the hive, and they've got about 30 minutes to do that. So does it benefit drones to spend all their time flying to DCAs and flying back, or spend all of their time at DCAs? At DCAs, because the longer they spend at a DCA, the more likely they are to encounter a queen. So drones typically, again, I'm overgeneralizing, this is biology, biology is messy. Drones typically stay at closer DCAs because they would rather be at one than go to one and come back. You're all with me on that. So they want to do this about 6 or 7 times during their 3 to 4 hour block of the day that they are going to these DCAs and coming back. And again, I rounded everything off to make it easier to discuss. But it's interesting because it's not perfect science. When we were looking for DCAs, we were told that drones start making DCAs between noon and about 3 in the afternoon. We couldn't find any DCAs. We thought our method was flawed. And then I just said, hey, why don't you just stay out a little longer one day and see what happens? And guess what time our DCAs form? 4. So it slides a little bit, but where we were in Florida, we were getting DCAs later in the day. It's not necessarily perfect all the time, but we were seeing that, and that's what was working for us. Now, let's talk about it, why this is important. Well, you get DCAs occurring at certain heights, you get DCAs occurring at certain locations, and you get DCAs occurring at certain times of the day. I don't have these labeled, so I'm going to guess that this is dorsata. I think that this is florea and andreniformis, and I believe that is serrana, but I am totally just guessing because they're not labeled. These are different species of apis. One of the things that potentially led to the different species of apis is that these species that co-occur in some parts of Asia fly to DCAs at different times of the day, at different heights, and have different anatomy, not allowing them to cross with the wrong queen. Remember, a lot of these other apis species co-occur. So what makes them species? Well, again, it's a lot of things that happen at DCAs, and even some subspecies of Apis mellifera might do the same thing, where there's different times of the day, different heights, different characteristics that they are looking for. So what are some of those characteristics? Well, this is not a perfect science. This is something that needs to be studied further, but my colleague Nico and Gudrun Koeniger were using terms like depletion of the horizon. When a drone flies out of a hive, what is it looking for to form a DCA? And they would argue, well, if you stand in a valley and you've got a row of mountains and a row of mountains, at some point they are going to come together in a V. The horizon is depleted. There's a depletion in the horizon. So they would argue that DCAs often form in the direction of horizon changes. The only problem with that is in Florida we don't have mountains. So queens would never be able to mate. So my student was looking instead at some other things. She would use Google Earth to predict where DCAs might form and then go to them to see if they were there. And what she used as the prediction were changes in topography. For us, that means tree lines. When bee colonies are located in a field and they're flying to the edge of a forest and it goes up, we typically find DCAs along the edges of those fields or on the edges of any changes in topography. So this is one of those things that needs to be worked out in much greater detail, but for now, these are the best things that we can say. I know some of the interesting things about DCAs is DCAs tend to be relatively constant. Where the DCAs are this year, they'll be next year, and they'll be next year. If you put bees here and they go to DCAs and you map them all, and then you take out all the bees and bring back bees 10 years from now, assuming nothing major changes in the environment, those colonies will go to roughly the same spots. So there's clearly something that drones and queens cue into when they are choosing where to go as a part of finding these DCAs. And if you're looking for a PhD, that would be a great thing to study. All right, so remember back to the idea of energy in the tank. Queens also have about 30 minutes of energy in the tank. But queens do not need much time at DCAs. So they spend more time going to one and coming back. So that generally, again, biology's messy, but that generally will push them further away from the hives than the DCAs that their drones are using. There's some debate about this, but I'm kind of overgeneralizing something that's really messy. All right? So queens don't need as much time at DCA, so they tend to fly to further ones, as some data suggests, than the drones will. So drones generally will hang out within about a half a kilometer to a kilometer, whereas queens typically go further. And they think this helps decrease the chances of encounters, sexual encounters between related individuals. Again, it's messy. If you live on an island, these kinds of things will absolutely happen. If there are no colonies, no DCAs out here, then she's just going to mate with the drones who are available, right? But in general, it is believed that queens go to further DCAs than drones, and that's how they can help avoid inbreeding. I've talked to you already a little bit about how we study DCAs using this weather balloon system and being able to collect drones. This is a really good way of capturing drones, finding out about the genetics, the number of colonies available in the environment, etc. So now let's talk about the actual docking process. So queens go cruising into this DCA where there's this loose assortment of drones flying in the air. The presence of queen pheromone streaking through a DCA calls those drones together to form what we often call drone comets, right? The coalition of these drones that are chasing these virgin queens that are flying through the area. And what happens is when a drone catches the queen, he will grab her with her legs and he will begin the eversion process. Remember, that paralyzes him. So in the docking process, he often falls over backwards, paralyzed, and she is assumed to use muscles in her abdomen to continue the eversion process into her. Now I want you to think about that for just a second. Maybe this isn't as amazing to you as it is to me. But while flying, a male catches her, mounts her, everts, falls over backwards paralyzed while she continues cruising through the air, doing all the work. And when she's done, she releases him to the ground where the ants feast. That's pretty incredible, right? And when this happens, the drone will fall off and actually leave some tissue behind. We call this the mating sign. And this mating sign confused beekeepers forever, beekeepers and bee scientists, because we knew queens could not remove that themselves, so we assumed they had to fly back to the nest, have it removed by workers, and then they would go take subsequent mating flights. But Nico and Grugen-Kruniger and others have demonstrated that that's not the case. What actually happens is, remember back to that slide where I show you the endophallus in all of its glory, and I mentioned kind of a hairy patch, that Brillo pad area? Nico and Grugen-Kruniger assumed that in the docking process of the next drone, that Brillo pad type structure removes the mating sign from the previous drone while the next drone is subsequently everting into the queen. That's kind of crazy too. And they have shown through research that mating sign actually makes it easier for the next drone to find and dock successfully with the queen. Now that's crazy biologically, because why would one man help the next man inseminate the woman that he's trying to have as many offspring with as possible? But that's the power of polyandry. Polyandry is so important that any drone benefits by the queen mating with multiple males, because the colony's fitness is going to improve if she mates with multiple males. So any drone is going to have a greater likelihood of siring offspring if the queen mates with multiple males. So they had theorized that mating sign actually helps this process and might be, for lack of a better term, by design, to make it easier for the next drone to find. So when the queen is finished mating, she'll go back and have that mating sign removed. But what do queens like? Queens like days that are 20 degrees or above. I'm assuming that happens at some point here, right? Does it ever get above 20 degrees here? Maybe, I don't know. Her mating flights tend to overlap with those of drones, otherwise we'd be in trouble, right? And they are risky for queens. There can be high mortality when they leave the hive. There are insects that can eat them, dragonflies. There are birds that can eat them. The cold UK wind can blow them away, right? There's lots of things that can happen while queens are on mating flight, so it's quite risky. So when they go cruising through this area, they get this comet of drone chasing them. The drones almost wait in line as a cue. The process, it happens the exact way I told you. The male catches her. You can see the tip of the abdomen is open. The queen opens her abdomen to receive the male. He starts the eversion process, which I've described. And while she is flying with his dangling body, she collects the semen from his endophallus, and it goes into her body where she stores it, not yet in the Spermatheca, but in the median and lateral oviducts. Stay tuned. That's important. All right. So when she's done, she flies back to the nest with that mating sign. Assuming it all goes well, she'll mate with somewhere in the neighborhood of 11 to 17 drones. Last week in our Entomology and Nematology Department at the University of Florida, we had Dr. David Tarpey, who's a professor at NC State University, come and give our departmental seminar. And he has done a lot of research on polyandry in queens and found that the average queen mates with about 15 males. That's about the target, as it were. So it can be much higher, it can be lower, but usually 15-ish is where you end up. And there are lots of benefits of polyandry. He talks about those. Keith Delaplane has studied that. Lots of people have studied that. You get more brood, increased sperm storage, better longevity for the queen. Pheromones, we know colonies benefit when queens mate with multiple males. And when all is said and done, the queen will return to the nest with the mating sign in place, and the workers will remove it. Now the queen has an interesting problem. Before I get there, I want to just say one quick thing. As a result of this, queens usually take one, maybe two mating flights, but it's usually very, very low numbers, usually one. Which again is contrary to what we thought years ago when they thought they had to have this removed by workers. All right, so there are lots of factors that can lead to successful or failed matings. Weather, age of the queen, temperature, wind, cloud cover, all kinds of these things. The number of available drones, lots of things can impact the success of the copulation. And drones, we know, die shortly afterwards. But before you feel sorry for them, the vast majority of drones will never accomplish their life purpose. If a colony is producing 2,500 drones for every one queen they produce, and queens mate with 15 males, that means the average colony is producing 24, I'm getting there, right? Not very many, right? Most of them don't ever make it. So these big fuzzy guys that look cute and dumb don't fulfill their life goal. And you know the result of that at the end of fall is the workers will kick them out of the nest, which is really remarkable to think about, right? There's this great picture we have from someone where there's this pile of drones on the outside of the nest. I think it's like 13 drones, and there's one worker. It took one worker to keep 13 drones from going back into the nest. So you can see the power of worker bees in this nest. So drones that don't mate end up living 3 to 5 weeks, typically after emerging, and that's too bad for them. Queens that do mate successfully, though, when they have the semen in them, they begin to develop further. You get changes on the molecular, physiological, and behavioral levels. She starts becoming a different bee than the one that left the nest for the purpose of mating. And so what happens? Well, remember, I told you the queen doesn't put that sperm directly into their spermatheca. To me, this is one of the most amazing things about the mating process. I'm going to average things to make it easier to discuss, but the average spermatheca can hold the volume of semen produced by one male. It's about 1.5 males, but to make it easier, I'm going to say one. So the average spermatheca can hold the semen produced by one male. So let's just say, for the sake of argument, this queen mated with 10 males. That means she's received 10 times more sperm than she can hold. So remember that Y-shaped lateral and median oviducts. That's where all the semen is. And for the next 48 hours, she uses muscles in her abdomen to fill her spermatheca with semen, with spermatozoa that she has in those oviducts. When she has finished filling the spermatheca, she flushes out the rest. So the vast majority of semen that queens collect, they'll never use because they flush it from the body, which is what all that says. So any given drone, based on drone math, might only have about 3 to 5% of the sperm that he gives her make it into the spermatheca. And in the spermatheca, you've got weirdness happening as well. A lot of people will ask, does that sperm mix thoroughly in the spermatheca? Well, the answer is yes, but eventually. Does it matter if you were the first or last drone? It does not matter. Are all drones represented equally in the spermatheca? No. And what's funny is some other research has shown is it doesn't really matter how many drones a queen mates with. About 8 drone lines make up the dominant patrolines for the eggs that are laid in the nest. To make things even more confusing, when queens lay eggs that workers eventually choose to become future queens, they tend according to research, to choose the less represented patrolines, which has led some people to think there's royal patrolines, which I think you have here in the UK, right? We did away with that a couple hundred years ago. You guys are keeping it around, but what? To each his own, right? All right, so you get these bloodlines, these royal bloodlines. So it's fascinating what happens in the Spermatheca. And this dictates everything that happens in your colonies. It's crazy, crazy, crazy. So remember, a well-filled Spermatheca will look like this. And then, depending on the life of the queen, she'll be able to lay eggs and fertilize eggs from that pool of semen that she collects, whether she lives months or years. Now, let's talk a little bit about some further development because I really want to get to a last slide and spend a second or two on it before I run out of time. We know that when queens have received the semen, when they put it into their Spermatheca, and they become sexually unreceptive, they're no longer attractive to other males, they will have their ovaries start to develop even further, they'll cease taking mating flights, and they begin to lay eggs. So this mating behavior triggers a lot of physiological changes in the queen, which makes her a mature adult queen capable of laying eggs. We see again the fleshing out of those ovaries. They get much larger. Her abdomen goes from kind of small, runny virgin queen to much bigger queen. There's a lot of production of new proteins like vitellogenin and all kinds of other things. Queens essentially become queens after the mating process. That is when they are capable of laying eggs. That is when they attract the retinue of workers that we know queens are so famous for. That is when they start conferring genetic traits to their offspring that will dictate the behavior, the biology, the ecology of the individuals that they produce. We know some things that cause these change, but we don't know all of them. For example, how are these changes triggered? Is it the volume of semen? Is it the number of times that they mate? There's been lots of research on this through instrumental insemination to see what triggers this, and honestly, they're still trying to figure it out, how this post-sexual maturation happens. But what I want to spend time on, is I've got about a minute and a half left, is this idea of poorly mated queens. In the U.S., one of the chief complaints about colony losses is that queens seem to not last as long. Okay, think back to what I told you very early in this lecture. The idea that queens aren't releasing a number of sperm, they're releasing a volume of semen. Tarpey and others have shown that as queens age, think about that Spermatheca. Queens don't know the quality of the sperm in it. They'll lay an egg, they'll stick their head into a worker cell. If it's worker size, they turn around to put in a female egg. Queens pass it down the chute, they release that volume, and they think they've laid a fertilized egg. But what they've shown is that as a queen ages, more and more of those spermatozoa die. So queens think they are laying fertilized eggs, but workers can detect that increasing proportions of those eggs are male. And when the queens cross a threshold of male producers, the workers start the supersedure process. So one of the things that they believe lead to shortened lifespan has nothing to do with the quality of the queen, but everything to do with the quality of the semen that they receive. A last point about this is that in the U.S., shipping queens is very common. Just to give you one example of how this could be impacted, queens undergo huge temperature fluctuations while they are shipped in an airplane from Georgia to Washington. And those temperature fluctuations have been shown to lead to lower quality and higher mortality of the sperm remaining in the Spermatheca. So by the time they purchase a queen, the life expectancy is triggered. So lots of things that happen in the Spermatheca can lead to failing queens. If I've done my job, we've talked all about sexual maturation in queens and drones, about DCAs, how polyandry is good for bees, the steps of copulation, all of these things, and how honeybee queens mature after they mate. If you have questions for me, you can follow these QR codes. That's our lab's website. This is our podcast. That is my email address. If you're interested in following us on any social media accounts, You can find us at UF Honeybee Lab. I'm going to leave this up for 10 seconds because as I turn it back over to Norman, I'm going to put my final thank you slide back up. So about 10 more seconds to take a picture of the slide. And then I'm going to thank you so much for your attention. It's been a pleasure talking to you about this topic.
Right, thank you very much, Jamie. That was great. A lot of ground covered. I'm sure there are time for just a few questions, though, so who's going to be first?
Make it easy. I'm old enough to feel comfortable saying I don't know.
Hi, Jamie. Thanks for a great lecture. One question. I've read various accounts of why queens become unable to go and mate eventually if they're held in the colony and can't get out. What physiological changes actually prevent them going out or prevent them from properly mating?
So if I understand your question correctly, there are times you're saying queens won't go on mating flights.
If you have bad weather and the
Yes.
queens don't get out, eventually they can't mate.
That's correct.
Why is that?
So if you remember a couple slides ago, I was showing the instrumental insemination process. People don't know for sure. They are trying to figure out those cues that cause queens to have passed the threshold, so to speak. When you instrumentally inseminate queens, you collect semen from drones with a needle and put it into a queen. Those queens never go on mating flights, but start laying eggs. So something in their mind has been triggered to make her cross that threshold. And the only thing that they could figure in that context is either the reception of sperm or somehow the CO2 that they use to knock the queen out while this is all happening. So the point of that slide a couple of slides ago is we've not figured out yet what makes it where queens go, oh, I'm too old to go on a mating flight, or I've done everything I need to do, now I'm going to start laying eggs. It's still a work in progress. That's a great question.
Thank you. Can I ask you about capensis? You tell me
You
that
may.
worker capensis lay female eggs. I've been told recently in South Africa that those eggs, if they're fed correctly, can become fully functioning queens.
Whew. All right, yes. So that's the question.
Is that
All right,
true?
all right. So I love Apis mellifera capensis. I love this bee. It's fantastic. So the question is if a worker lays a unfertilized yet female egg, can it become a queen? Yes, it can become a fully functioning queen. The interesting thing is, though, is that when workers begin laying in colonies of Cape bees, laying workers, they take on the pheromonal bouquet of queens. So the colony thinks it has a queen, so they're not triggered to produce one. But you can produce queen offspring from the eggs laid by capensis workers that are unfertilized. It's fascinating. Bees are fascinating. I hope you appreciate that. If for nothing else, I hope this talk makes you go, gosh, what we do is cool. All right.
Jamie,
Yes,
there's
sir.
one thing you didn't mention in all this, which I understand underlines the importance of genetic diversity in a colony, and that is the rate of crossovers during meiosis that occurs in honeybees, which are supposed to be reputed to be 10 times greater than that in the human reproduction.
Sure. So his comment is the rate of crossover in strands of DNA in honeybees is 10 times that of human. It's possible. I've never heard that. But I will tell you the crossover rates are very high in honeybees. And, polyandry by itself and the mixing is just really incredible, the genetic diversity within a colony. I will tell you, I'm going to chase a small rabbit here. I've done a lot of work in South Africa. That's where I did my PhD, and we continue to have research projects there. We were looking at samples of bees a couple of years ago where the estimates that the queens that the diversity within a single honeybee colony suggested that there were more than 50 males that queen mated with. So you can get incredibly high mating rates, you can get incredibly high crossover rates, the genetic diversity of work, it's just incredible, which is why when you've got restricted flights and poor genetic diversity that we have all of these problems with mating and the quality of colonies and things like that.
Hello, Jamie. So
Hello.
I'm over here. Hiya.
Yes. Hey.
So the one question I've got is, how do the queens and the drones know where to go? Is there
I-
any research
yes.
as to how do they know where to go?
The question is where do queens and drones know where to go? The only theory I've ever heard was that depletion in the horizon theory, that they come out of the hive and just fly towards that. And again, I was making the argument that in Florida we don't have those valley systems. So it seems, I will say before I answer this question, this is one of the gaping holes of honeybee sex research. But it seems that they will hit, at least from our perspective, these kind of changes in topography, and that's where the drones hang out. Queens, on the other hand, they probably are queuing into the same thing, and when they start getting drone activity, they don't have to go further. Another thing that's interesting is that while you get DCAs, while you get drones going half a kilometer to a kilometer from their hive, they can form DCAs much quicker. If you start one of those balloons at a functioning apiary and walk away, it's not long before you have drones chasing you. So it doesn't even have to happen out there. So some people suggest, why don't drones just hang around the front of hives? Because they catch queens going out. Well, who are those drones likely to be? The ones from those hives. And so, but that is I mean, it sounds like we know a lot when I talk about it, but I've told you what we know, right? So everything else is a big gaping hole that's waiting to be discovered.
Right, this would probably better be the last question over there, Derek. Then.
This is a complementary question to the previous one.
Sure.
That was the where, and this is the when. And does the actual time of mating vary with latitude? Are the bees dividing up the amount of sky to the horizon, or is it to where the sun sets?
Sure.
So if it was, one would expect it to vary as you went north, as you went north and the latitude changed.
Yep.
Has anything been done on that?
So the answer is I don't know, but if you think about my German and my Florida example, Germany's at a higher north latitude, and they're telling us that we need to look for DCAs between 1 and 4. And Florida's at a much more southern latitude, and we were looking for DCAs between 4 and 7. So it was clearly affected in our case by latitude. We were getting very different times based on where the bees were in the world, right? And so I think a lot of those things will dictate where and when DCAs form and when queens and drones will go to them. I have no doubt. That would be my strong hypothesis, in fact, for it. All right, you guys have been a great audience. Thank you so much. I appreciate it, Norman.